All right, let's lock in, fam.
Could always just use my name, Jordan, something that we hear a lot from our guests.
Yeah, is that better?
No, should never use my name.
Welcome to Absolute Genius, a podcast series from Thermo Fisher Scientific. I'm Lisa Crawford.
And I'm Jordan Ruggieri, and today we are diving into integration site analysis and how CROs like ProtaGene are shaping this field.
Our geniuses today are Michael Stump and Matteo Franco, president and lead scientist respectively at ProtaGene. They share insights on genotoxicity, how they use digital PCR, and the balance between regulatory expectations and customer program goals.
We hope you enjoy the conversation. Michael and Matteo, thank you so much for joining us on today's episode of Absolute Genius. Could you maybe start with just giving a background about yourself and a little bit about ProtaGene as well?
Sure. Thank you, Jordan. It's nice to have us. My name is Michael Stump. I've been the president of ProtaGene for about 18 months, but in the industry about 25 years. ProtaGene is a small CRO that really focuses on two main areas. We focus on CMC, mass spec design, and then also on the cell and gene therapy side, we focus on the ISA, integrated site analysis.
I'm Matteo Franco. Nice to meet you. I'm the lead scientist here at the Burlington site. I've been mostly dealing with integration site analysis, but I've been with the company since 2018. I moved to the US when we decided to open a site here, and I've been here since working on mostly on integration site analysis and molecular BioA capabilities. That's excellent. Yeah. Can you talk a little bit about what is integration site analysis and what are some of the considerations for customers that might be looking to do that? Of course. Yeah. So integration site analysis is a field that is has become more and more relevant the more viral vectors came into play into the gene therapy field. It's basically looking at the genotoxicity of these viral vectors when they're administered. Our job here is to basically get the samples, sequence them, and then determine where the integrations happen. So, for instance, in case of a lentiviral vector, where you have an active integration mechanism, you'll find a lot of integration sites. So you want to make sure that they integrate into the genome in safe spots. Do not activate any oncogenes or the likes. So basically, that's a metric that helps you during your clinical trial to determine whether your therapy is safe or not. Interesting. I guess we've never, or at least I haven't thought about this. We do and talk a lot about the vectors themselves and creating them and getting them in the virus, but once you actually get past the quality and some of the the look at the vector and the virus itself, actually what happens within the subject, seems like it would be a really critical area to dive into and make sure that you have an understanding. I mean, is it just looking at those integration sites, just making sure that it's not incorporating in areas you don't want it to, or are there other aspects that you look at once it actually gets into a subject and kind of post that initial manufacturing and kind of safety check into your maybe early phase clinical research? So the main readout out of the integration site analysis is the location where the integration happens, but also the relative frequency. So in terms of like all the reads that we cover from the subject, how many are integrating there? So you're then able to establish whether you have a so-called clonal expansion. For instance, a single integration event then led to the multiplication of cells, and that could be linked to a problem. It's not a direct connection by any means, but it could be a warning. But we're talking about sequencing, so you have a lot of data that you can dig deeper into. So you can look at which part of the vector integrated. Normally, length is integrated as a whole, but AAV is prone to recombination, so it can be any part of the vector really. So, for instance, you don't want to have a promoter that integrates near an oncogene. That's one thing that you can check and make sure that it doesn't happen. You can just also in terms of like topological integrations, you can see what is the distribution across chromosomes. You can see the distribution nearby cancer genes if there's an integration hotspot.
that it's typical for your vector. So there's a lot of other interesting quality attributes you can search. I mean, it's still early stages, but like people have started to delve into a long read, for instance, integration site analysis. So that gives you a better idea of the integrity of the vector when it integrates, or if it persists episomally, how it persists. There's a lot of data that you can extract out of these.
Taking maybe a step back even and talking about ProtaGene and your approach, can you talk a little bit about maybe what a CRO is and some maybe considerations for maybe some of your potential customers that might be looking to have this research done? What exactly do you do to help make sure some of these things turn out well or as best as you can expect?
So a CR is an interesting term. A CRO is a contract research organization. And somehow that gets lumped in the industry. Sometimes CDMOs are included that with the manufacturing piece as well as the testing piece. So within the CRO world, ProtaGene is basically an advanced testing platform. So we test both at the early phase and late phase. So from CROs, they can be one-stop shops where they can run a whole suite of assays for a lot release, or they can do early phase for design and different pieces along the way. ProtaGene fits into that puzzle where we're what I would call a niche CRO. And by niche, I mean we only offer some of the assays you would need for maybe a lot release, but we really focuses on advanced characterization. So if you come to us for basically on the CMC side, we could be there for early phase and characterize, but we can also support late phase and GMP for lot release and advanced stability studies. On the cell and gene therapy side, same thing again. We can be there in the early days to start the assays to show if there's any issues up front. And then we can go all the way through commercial phases and help on the back end as well for lot release. And in fact, we actually are just becoming a CLIA lab both in the US and Europe to support CLIA for ISA, which is very new. We're supporting some of the first companies that have done this in the industry.
So help me understand, is there a certain kind of viral vectors or a certain product or a certain disease area that people are more likely to use your services? Or is this something that kind of applies to everyone in the space, depending on, their interest in going deeper?
So normally, yeah, our customers are more into the gene therapy as a whole. We've seen mostly viral vectors, a lot of lentiviral vectors, a lot of AAV, a lot of retroviral vectors. It also depends on the trend of the industry. We've seen a lot of LNPs popping up lately. At the end of the day, we try to evolve with the market as well, right?
Yeah, so basically it comes down to what do you need? Do you need to support lot release or do you need characterization for early phase or advanced characterization for filing? So for us, we could do both. So the CMC side really does the characterization piece, which is more of a nice to have. In some cases, it's a need to have in other cases. The ISA piece that we do, and that's kind of an interesting part of the industry right now. For us, if you want genotoxicity, this is the best way to do it. And still in the industry, it's a nice to have, not a need to have. But if you have a severe adverse event, then it becomes a need to have. So that's kind of where we're at. So upfront, some clients are looking at a risk mitigation strategy where we develop the assays and we're ready for the severe adverse events. And in some cases, they do test about 10% of samples just to be safe.
Can you elaborate a little bit on genotoxicity? What is that, and how could it impact some of these developing therapies?
Yeah, of course. So, in terms of genotoxicity, we mean toxicity for the subject as a whole arising from a genomic level. This is where integration site analysis comes into play. We make sure that like the integrations of these therapeutical vectors don't impact the subject in any kind of way. So, for instance, if you have a lentiviral vector integrates multiple times into the genome, after sequencing, we determine where this integrates and if there's a potential for a severe adverse event. Or vice versa, if we have a severe adverse event and we want to make sure that it was not the vector's fault to get this result. So again, we use integration set analysis to determine is it the vector that activated this oncogene or was it something else?
How does ProtaGene look at, let's say, technology acquisition, right? So you have your kind of set protocols and some of your methods that you support. How do you evaluate when there's a new technology that you should integrate in, a new method, or you need to bring in some type of new equipment to support customer needs?
there's kind of a dichotomy of differences in the industry. So from the FDA point of view and the regulatory agencies, they want something that's old, tried, and true. They don't care how old it is. If it's got 20 or 30 years of history, fantastic. I would say in the cell and gene therapy space in the last few years, it's kind of the opposite of that. Whatever's brand new and shiny, they want it instantly. So it basically comes down when you're looking from an R&D perspective, especially in the new cell and gene therapy space, and even large molecules and ASCs, they want the latest, greatest instruments that come out in the market, which is what like Thermo and other companies support. For a CRO, we're kind of stuck in between, right? It really is we want to use the platforms that are most important to our clients and to support the science. So from a CRO point of view, we try to be agnostic. We use whatever works best. So what usually happens for us is when some new technology comes out to the market that's getting a lot of buzz, until our clients we can show that more than two or three need it, then you don't make the investment up front. So we have a technology council that whenever our either our BD representatives or we send the scientists to conferences where they'll come back and they'll say, "Hey, we saw this new piece of equipment in Thermo that can do X, Y, and Z." So then we'll take a look at that. And as soon as we have two or three clients that say, "Hey, we really want this," and we get to the point where we get to contract and it works, then we'll look to make the acquisition. The good side is we might be slow to adopt, but usually in our space, why have one when you get two at twice the price is the motto. So usually in CRO world, you always buy two or more because if one breaks, you can't ever afford to be down. So we might be slow to adapt. Then when we do, you usually invest pretty heavily in the space where it's applicable.
No, it makes sense, and we've had conversations like this before as well. Whereas the FDA, like you said, tried true 20, 30 years. They want something that's there, but that's not always where the actual teams doing the research live in terms of trying to figure out new methods or new ways or new approaches to make sure targets are safe or try something new, right? And be kind of on the forerunners of innovation for the space. So it's always a balance. It sounds like a really good balancing act that you have with an acquisition team and a technology team and looking at customer need. How do you approach looking at these different technologies? So say like, we love PCR, we love qPCR, dPCR here at Absolute Genius as well. How are those technologies utilized in your space?
There are definitely a lot of different ways that we can utilize them. That also ties into the previous question, I think, because like, sometimes you don't need a new instrument to perform a new assay, right? So I feel like, especially now in with digital PCRs, there's always something new that people develop. For instance, I think that I came across recently was then it also gained a lot of traction, ASGCT, was the use of dPCR to assess the integrity of a vector. Because now with dPCR is very easy to multiplex. A lot of people are starting to experiment, with the multiple targets that you can use. So you can use multiple primer probe sets that span across the vector and then assess the integrity from there. You can use them for vector copy number. We use them for replication competency, biodistribution, transgene expression. Like they're very very versatile, and the multiplexing capabilities also in general like simplify our work. They make it way easier, and it gives you a better readout at the end of the day, because back in the day, at least most of the BCN assays we were used to do on qPCR were a single target, when now we can just throw a reference in there and it's super easy to do.
That's cool to hear. I mean, it sounds like between the different technologies, you kind of use them to answer different levels of questions that you or your clients might have, but also in new ways to make things a little bit easier, faster, maybe less expensive, and on everybody's end. So rather having to maybe send something through to sequencing, where might take a little bit longer or be a little bit more expensive, you can use something like digital PCR for that, making sure that it's the right vector and just using it in different ways, right, to help make sure that the questions are answered properly and with enough confidence to keep things moving forward. Is that Is that accurate? An accurate statement?
Very, very. Actually, that's a very good point because they can also be used as an orthogonal method to sequencing. Like, do we want to really make sure that an integration site really happened at that place? And we want to know exactly the quantity because maybe sequencing, is still a relative metric, right? So we want to know exactly how many copies you have. Confirmatory dPCR, why not? Same thing with gene editing. Do you want to make sure that a certain edit happened on a certain site? You can also throw it on the dPCR. So it's a very powerful instrument for us, and um, as you said, can be used in a lot of different ways, also to complement what we already offer.
When a client comes to you and they say, I have XYZ problem or I need to get XYZ done, how much of what you guys do in response is like pretty cut and dry or something they ask for? And how much of it is problem solving, like you just said, trying to integrate these different technologies?
That's a great question. And to answer that, there's a wide variety. So I think from the CRO world, I think to answer your question, I'm going to probably take a different approach and then I'll answer in the back end. So I think there's about 4 or 5 factors that people look at when they select a CRO. One of them is what is the quality that you need to submit to the agencies to check a box. The next one is the science really strong in supporting that? The next piece is probably turnaround time. How fast can you turn this around? And then communication is also a piece. How well do you support this so we know what's going on? And the other one is price. So you kind of blend that all together, and then it depends on who is asking that question because we work from everybody from large pharma all the way to a startup right out of the university. So depending on who it is, they're going to have different levels of expertise. So it's not to give a hard time to the startups, but typically they don't always know the whole process or whatever CRO does. So when they come to us, they think they have an idea of a problem. And in that case, CROs, we really do spend a lot of our time of coaching a team and explaining to them both the regulatory piece, the science piece, and everything we do and everything in between. We have to do a lot of hand holding there, which is what we like to do. We enjoy that. When it gets to some of the larger companies, the larger pharma that have been at this game for a long time, they pretty know much exactly what they want. And in some cases, we're even pushing the science together in partnerships where we're taking the technology and we think we have it figured out and we'll do something new together. And then we publish. So it really depends on the audience and who's asking the questions. But overall, this year, we do spend probably half our time coaching clients of what the science is and how to best integrate that. And there's also a fine line there with quality too. As a CRO, we're not trying to tell you how to file your quality reports. That's up to you. What we really do is just try and show you what we think will work best in any given situation.
What do you see as what do you think this field looks like in the next couple years? What's exciting? What's coming up and is really something that you're really eager to get into the lab or get into work and keep pushing forward?
So I would say in a previous life before I joined ProtaGene, I've worked in the cell and gene therapy at a very large group CRO, and I've seen this very well. So I think there's very much challenges these days in the cell and gene therapy space. I think everybody rushed out and thought we can solve a lot of problems, and it's really unfortunate because at the last ACGT conference, it was well known that everybody was starting to celebrate some significant wins. Like we're really starting to see the wins, but we never asked the original question: is how are we going to pay for this? And because that question was the elephant in the room, it's like everybody kind of knew it was going to hit. I think 2024, late 24, 2025 was the year where everybody that we couldn't avoid that question anymore. So the cell and gene therapy space is really, really going to be challenging right now. I think everybody's looking at lipid nanoparticles and different things of what they can do to get away from the different viral vectors. But unfortunately, there's enough in the pipeline with viral vectors that we really have to play some of this out. Overall, with ProtaGene, we would love to be able to support, the genotoxicity and advanced platforms for this cell and gene therapy space. But like I said, it's really challenged. Overall, that said, I think also on the ADC side and large molecules and mAbs, there is a ton of resurgence in there because as the cell and gene therapy has dipped down, now you're seeing this influx now with ADCs and AOCs and different modalities coming back into play to fill that gap and go there. So from our point of view, as CRO, we are agnostic. Our CMC side can support either modality. ISA is favored more to cell and gene therapy, but we have enough assays to balance out what's going on in the market. But over the market itself, right now, it really is a shame the cell and gene therapy space. With all the great wins we have, I just wish we could figure out the payer piece.
That's a very good point, and I think in terms of like science, like our key will be to be ready to adapt to whatever comes next, because as we mentioned earlier, like LMPs are becoming more and more central. So we need to adapt and provide basically analyses that can better fit those kind of vectors. That's pretty much it. Like in general, molecular bioanalytics and sequencing will still be essential. We will just need basically to adapt the current methods to better fit the LMPs, especially in terms of like sequencing. But yeah, in terms of molecular bio, we're basically pretty much there.
Hey, Jordan, I've noticed that we've had researchers talk a lot about wanting the freedom to explore their biology without workflow limitations.
That is very true, and that's the idea behind the Freedom of Expression Gene Expression campaign from Thermo Fisher Scientific, giving scientists flexible, scalable solutions to confidently measure gene expression their way.
Whether you prefer probe-based assays like the Applied Biosystems TaqMan, dye-based detection like the Applied Biosystems CyberGreen, or need solutions that support high throughput or targeted verification workflows, there's an option designed to fit your research needs.
It's about enabling flexibility without compromising performance, so you can focus on uncovering meaningful biological insights.
To learn more about the Freedom of Expression campaign and available gene expression solutions, visit thermofisher.com/freedom-of-expression. And be sure to check out our latest promotions at thermofisher.com/promotions. Products are for research use only, not for use in diagnostic procedures.
And now, let's get back to our conversation.
All right, I'll pass you over to Lisa for some career background and career corner time.
Yes, we're going to get away from a little bit of the science and talk more about you guys and both your backgrounds. I actually have a question for both of you that I haven't asked anyone yet, and it occurred to me because I think the space, especially, CRO, CDMO, if you're not in that industry, I think it can be pretty opaque for people to understand kind of how it all works, right? So I was wondering if both of you separately, can kind of walk me through what a typical day looks like for each of you, since you're also in such different positions. Just what it is that you come into work, an average day, what kinds of things do you do?
It's hard to answer in my opinion because it really depends also on the like we rely a lot on the client interaction, right? So um, one day can look very different from the other. But in general, most of my days are spent uh, in internal meetings as as per usual, where we align on certain like projects or in my case also developments because usually I'm also involved in those. And then that's usually what happens in the morning because we also interface with our German side, so that's when usually that we manage to overlap a little bit. Another part of my day is definitely working on the client projects, handling communication with them, preparing results, slides, reports, instructing the lab on what to do, any planning that it might require or and anything. And then I also am involved in sometimes in the BD calls where where I'm there as a subject matter expert to support our BDs in getting more clients, managing current ones as well.
So are you ever in the lab or you're more of a supervisory strategic?
I'm more of a supervisor sometimes. I can end up in the lab, especially mostly for supervisor things, showing up there, check on certain projects, devices, or analyses. Um, and in rare cases, I'm also the one pipetting, but I usually keep that as a minimum.
When people are on vacation, maybe they got to get you in there.
Yeah. When you need like several operators, for instance, and some people are out and I need to step in, but it's more rare. I usually am more of an office worker, let's say.
Michael, what about you?
So basically, I've been in executive management for over 10 years now in an industry 20 plus. So in the executive management role, I would just say my job is a controlled, chaotic firefighter. That's all you do is put out fires. And I'm convinced people that do this for this longevity is we like the challenge. Because if I went to large pharma and worked in a similar role, I'd work on one or two molecules for 5 years. I think the reason people select this hero world is they love the variety. I would think in 1 year alone, I worked on 139 different compounds in a previous life, just to give you perspective. And so for people who choose this hero world, I think they really just love the variety and the action. And there's always just constantly stuff happening, and it's always a balance of timelines to get what we need to meet that and how to do the best science possible given the timelines. I think the hardest part in the CRO world is a lot of scientists coming out want to do perfect science. And at the end of the day, you have to balance that with science that is strong enough to meet a deadline. Doesn't mean that we'd ever, for 1 second, sacrifice the safety of any subjects. It's more along the lines of scientists tend to want to wait until everything's perfect when we already have an answer that checks the box and you're good to go. I think that's the biggest challenge I have in the CRO is convincing my scientists to we want to do the best science possible when we get there, but we can't overdo it either because the timelines and the costs just doesn't is prohibitive to that factor.
Another question that I like to ask people is if you were to run into someone at a conference or somewhere, younger, just starting to get into science in this industry, what is something that you maybe wish that you had known going into this that you would share with them, just to kind of guide them and maybe some advice for them as they get into this?
So to me, the way I explain it is if you want to get in this space, everybody wants to work in large pharma. And I would say is that doesn't, there is no clear path coming out of the university to work in large pharma anymore. You have to do your stint or pay your dues basically in the CRO world because the way the industry works right now is you don't have any experience and you really need to get that GMP. So my advice to the young kids is start working for a CRO and gain your GMP experience and learn the techniques because there's two pieces of what we do. There's the science piece and then there's the documentation of the science piece. And they're two very different things. And I can take a lot of really good scientists that are R&D scientists and can't do the documentation. So anybody that wants to get in this space, I would just figure out what you're really good at. I mean, if you want to do the science, then that's okay. Then you have to stay on the R&D side more. But if you really want to do the impact and help the subjects and the most you can, then you really have to figure out the GMP piece and learn the documentation, which is a whole different thing.
Can you explain GMP?
So there's different phases of the FDA approval process. There's preclinical, which is early phase. It doesn't need to be GMP. Some people say GXP because there's good laboratory practices, there's good scientific practices, and there's a couple other things in there too. So good clinical practices, GCP. There's everything. So GMP is the kind of the main one. Whenever you get to clinical phases, you have to have a manufacturing process that's aseptic and guaranteed. So you have to do things under GMP processes. So all CROs, unless you're in early phase, you may not deal with GMP, but for most of us, we have to deal with GMP. It's getting to be more and more.
Thank you for clarifying.
Yeah, sure, no problem.
Yeah. So Matteo, do you remember the question?
Yeah. No,
Okay.
I do, but I feel like I am little bit on extreme case because I'm still working for the company that I joined right after my studies. So, I mean, it ties a lot into what Mike said. I think you need to find what you're good at and or at least what you think you're good at, and then explore from there. Because, um, like academia and industry are very different words. You really need to figure out what you're best at. Like I have some friends that are like diehard academia people. They like the environment. They like writing grants. They like being in the lab the whole day. That's not me personally. So maybe people that are more like me that they want to have a more fast-paced job like client interaction or something like that. Then maybe the industry, especially a CRO, could be more of a better fit for them. So yeah, I would try to engage which is your main interest at the end.
That's all good advice. That's
So yeah, I think we just have one last set of questions, unless Jordan, there's anything else. We like to ask each person, what is a memory you have of maybe your most embarrassing moment when you were coming up through the ranks, maybe in the lab, maybe some other place, and then also, a crowning achievement or like a great memory that you have of something that you feel very proud of.
So an embarrassing and great memory.
We like to balance.
Oh, I have a lot of I have more embarrassing than great, so that's easy. I think for me, the most embarrassing one is I had just become director of a lab. We had some Japanese a company from Japan that came and they didn't speak any English, and my job was to tour them. And so I naturally talk very fast and I'm very animated. So when I went on the tour, I talked really slow and simple, but I didn't tell the lab this and they didn't know. And so they thought I was being degrading and racist to them and reported me to management. And then I had to sit down with a team and explain that they didn't speak English and they didn't know. So for me, I think that was probably the most embarrassing point is once the team understood and I explained that everything was fine, but it was a learning moment for me when you tour labs, you tell everybody what's going on and inform them. So I think that was kind of embarrassing. I think my greatest achievement working in the cell and gene therapy space, and there's probably two different stories, but very similar, is helping one company get the first in the market for solid state tumors and developing cures where you know that your survival rates went from people going to hospice that within 30 days, these people are not only survived hospice, but they have true cures and get back to their life within 2 months. And being part of those teams where you have a GMP run coming off at 10 o'clock at night on a Friday night, and you have to have 3 rounds of coverage, and you make sure the staff is there and they're able to get the a lot approved to have those outcomes, and just working through a process where that becomes somewhat seamless. It wasn't seamless, but it was a process that we developed. But just knowing that you worked on molecules that truly save people's lives is really hard to beat.
My great memories are more or less that's like I feel like the great thing about like working for a CRO is that I mean sometimes you get into anonymity because like sometimes you're providing a service, so some clients are like, okay, you provided the service, bye. But other times they share the results with you, and it does feel like what you do actually matters, right? Because you get notice that maybe the treatment was successful and you worked on the treatment. So you realize how much like it means, like what you do it means for maybe people that have like very bad disease, or you know they somehow manage to survive at the end, and so it makes you really proud of what you're doing. And in terms of embarrassing moments, I guess, I mean, I was very green back in the day, but I remember during my bachelor internship, I was part of the retroviral lab back in the University Hospital of Bologna. And we used to have a very old piece of equipment. It was a light cycler qPCR. It was so old that it still had like Windows 95. And on top of that, like nowadays, qPCRs are on a plate, super easy to handle. That was a light cycler, so it had glass capillaries. So what happened basically was like, I was being tutored, like they showed me how to do the qPCR, like be careful, capillaries break. Of course, no problem. So I go and get these samples, and they're like, oh, remember, this is the last of the sample we have. Be extra careful. Got it. Pipette reaction, run the cycling, get absolutely no results. Open the lid, everything completely broke.
Oh, gosh.
With all of the reaction at the bottom of the light cycle. So it was also a mess to clean. One of my first forays into the lab, and what was also probably one of the most embarrassing. But yeah, at least the staff had a good laugh. I didn't on the time.
When it's you, you go into the back and just have a moment to yourself after that.
Now looking back, I laugh at it, but
I love that question. It makes everybody seem human, right? We've had so many different examples of embarrassing things and things blowing up. I have a bunch in the lab too, and everybody is human. Everybody makes mistakes, and still wind up in great places, right? So it's always fun.
Yeah, the other one I could probably talk about is the when I lost a mouse.
I'm assuming you mean like a mouse, not
Yeah, mouse, an actual mouse with an animal studies. My wife and I had a company where we were doing an anthrax vaccine, and we had to inject 40 mice, and we had to bleed them first to get a baseline and then inject them. And on mouse 39, after 16 hours of that process with the last one, I go to bleed the mouse and it jumps out and escapes and it's running through the lab. And on my hands on my knees at 2 o'clock in the morning, trying to chase down the mouse. And it took me about 30 minutes to catch it before we finally could get the mouse.
Michael, Matteo, thank you so much again for this amazing conversation. I learned a ton and had a lot of laughs along the way. So thank you very much for your time, and we'll catch up soon.
Yeah, and just thank you for your time and your interest in ProtaGene and just our life story and how the CRO industry works. It was great fun. So appreciate you.
yeah, appreciate it so much. Thank you for the opportunity. It was very nice to talk to you guys.
That was Michael Stump and Matteo Franco, president and lead scientist respectively at ProtaGene in Burlington, Massachusetts. We've got more great conversations and new episodes dropping soon, so stay curious and we'll see you next time. This episode of Absolute Genius was produced by Sarah Briganti, Matt Ferris, and Matthew Stock.
Products mentioned in this episode are for research use only, not for use in diagnostic procedures.